You have trillions of cells in your body right now.
Oh, yeah, trillions.
And every single one of them, from the cells lining your stomach to the neurons firing in your brain, they all contain the exact same DNA. The text of the instruction manual is identical.
Right, which is wild when you really think about it.
It is. So how does a brain cell know to act like a brain cell and not, suddenly start beating like a heart cell? Today on The Deep Dive, our mission is to explore a truly groundbreaking paper hot off the presses from Nature.
Yeah, it's titled Gene Regulatory Landscape Dissected by Single-Cell 4-Omics Sequencing.
Exactly. We are unpacking how a revolutionary new technology, brilliantly named CHARM, is allowing scientists to read 4 entirely different layers of a single cell's instruction manual at the exact same time. Okay, let's unpack this because it's a massive leap forward in answering that fundamental mystery of cellular identity.
It really is. And if we connect this to the bigger picture, that identity relies on a system we're all pretty familiar with by now, which is the epigenome. Because we know that raw DNA is basically just an inert dictionary. It requires the physical and chemical layers sitting on top of it to dictate which genes are actually read.
Yeah, like if we use a library analogy to visualize the epigenome, the DNA is the text of the books.
I love that analogy.
Thanks. So the nucleosomes, the DNA wound around histones. Those are like the locked or unlocked bookshelves. And then you have histone modifications like methylation or acetylation.
Right. Those act like sticky notes telling the librarian to either ignore a book or read it immediately.
Exactly. Then there's chromatin accessibility, which is literally whether the aisle is physically open to walk down. Plus the 3D genome architecture, which is how the library folds in on itself. So a book on the first floor touches a book on the third floor.
And finally, the RNA expression is the actual reading of the book. But the challenge hasn't been understanding that all this exists. The challenge has been, actually measuring it.
Right, because up until this CHARM paper, the measurement process was incredibly fragmented, wasn't it?
Oh, completely.
Yeah.
If researchers wanted to look at accessibility, they had to run a specific assay, like ATAC-seq, on one batch of cells. If they wanted to map the histone sticky notes, they used a different tool, like CT and TAG, on a totally different batch. And mapping the 3D folding required yet another batch of cells entirely.
Which introduces the fatal flaw of multi-omics research, right? Batch effects.
Exactly. You're taking the structural map from cell A, the chemical map from cell B, and the RNA from cell C, and just sort of averaging them together. You lose the true interconnected reality of a single individual cell.
It's like trying to understand how a complex watch works by looking at the gears from 3 completely different watches.
That is exactly it. And that's where CHARM comes in. The acronym stands for chromatin conformation, histone modification, accessibility, and RNA expression multi-omics.
It's a mouthful, but it captures all 4 modalities simultaneously within the exact same cell.
Yeah, which is just mind-blowing from a chemistry perspective.
Yeah. Well, let's get into that. Because the secret weapon of the CHARM workflow is an enzyme called TN5 transposase. Now, naturally, TN5 is an enzyme that loves to seek out open, easily accessible stretches of DNA, cut them, and insert little tags.
Right, it naturally hunts for open space.
But you're saying the researchers use this exact same enzyme to also label the specific histone proteins, which are often wrapped up in tightly closed chromatin. How does an enzyme that wants open space get forced to tag closed structures?
So the protocol essentially tricks the enzyme by engineering how it's delivered. They use highly specific antibodies that bind tightly to the target histone, like a repressor mark, for example.
Okay.
Then they tether the TN5 enzyme directly to a secondary antibody that hunts down the first one. So instead of letting TN5 roam freely, they physically drag it to the exact histone they want and force it to drop its tag right there.
But wait, if you're throwing all these chemical reactions into one tiny cell, dragging TN5 to open DNA into closed histones, doesn't it just become a chaotic soup of identical tags? How do they avoid destroying the very data they are trying to read?
That is where the in silico, or digital separation, comes in. Before they even unleash the TN5, they assign highly unique barcode sequences to the different tagging complexes.
Oh, wow. So the TN5 that targets open chromatin gets one specific digital barcode.
Yep, and the TN5 tethered to the histone antibody gets a completely different barcode.
Ah, so they aren't physically separating the molecules in the lab anymore.
No, the physical molecules stay entirely mixed. They can flow sort the single cells into individual tubes and just amplify all the material in one single reaction.
And then the computer sorts it out later.
Exactly. When all that genetic soup is fed into a sequencing computer, the algorithm simply reads the barcodes and perfectly categorizes every fragment back to its proper biological layer.
That is just a phenomenally elegant solution.
It really is.
So let's do a quick recap of what we've covered so far. First, the epigenome has multiple layers: DNA folding, accessibility, histone marks, and RNA. Second, older methods measured these separately, missing the true interconnected picture.
Right, the batch effect problem.
And third, CHARM measures all four simultaneously in one cell using digital barcodes. Which leads me to a question for you, the listener. If you could put a tracking barcode on any biological process in your own body, what would you want to monitor?
Ooh, that's a good one. I'd probably track my sleep hormones.
Same. Now, to prove this wasn't just theoretical, they applied CHARM to mouse embryonic stem cells, right? And the data yield was staggering.
Yeah, out of 960 individual cells processed, 805 passed an intensely rigorous quality control check across all four layers simultaneously.
Which is wild.
The sheer volume of high-quality data per cell is unprecedented. In just a single cell, they detected a median of over 17,000 RNA molecules, 52,000 accessibility fragments, 103,000 histone fragments, and roughly 1.8 million 3D chromatin interactions.
Okay, let's ground those massive numbers in a real-world biological mechanism from the paper. They focus heavily on a gene called SOX2.
Right, the master regulator.
Exactly. SOX2 is a vital gene for keeping stem cells flexible or pluripotent. It keeps the cell's options open. When they ran CHARM on SOX2, they observed a very distinct 3D loop.
Yeah, the promoter region of the gene physically bent through three-dimensional space to connect with an enhancer region located really far down the linear DNA strand.
But the interesting wrinkle here is that the physical strength of that 3D loop didn't correlate perfectly with the amount of RNA produced.
And that finding actually challenges a long-standing assumption in genetics. We used to think that a stronger physical connection automatically equaled a higher volume of gene expression.
Like turning up a volume dial.
Right. But CHARM reveals that biological regulation isn't just a simple mechanical switch. The 3D loop is necessary, but it's just the architectural foundation. The transcription rate is fine-tuned by the histone marks and local accessibility within that loop.
It's a great contrast to another gene cluster they looked at, the Hox genes. If SOX2 is about keeping options open, Hox genes are the strict architects of the body plan.
They dictate exactly where an embryo grows a head and where it grows a tail.
Right. So in an early stem cell, a misfiring Hox gene would be catastrophic. The cell doesn't just turn it off, it buries it. And CHARM mapped a massive, dense blanket of a repressing histone mark, H3K27Me3, entirely smothering the Hox cluster.
The ability of CHARM to map that repressor blanket so clearly is a major technical triumph. Because remember earlier when we said TN5 naturally favors open chromatin?
Yeah, the TN5 bias.
Exactly. Historically, any tool relying on TN5 struggles to map repressed, tightly packed regions because the enzyme physically cannot infiltrate the dense chromatin.
Right. If the door to the library is deadbolted, the librarian can't slip a sticky note inside.
Perfect analogy. But because of the specific tethering method we discussed, CHARM proved it has incredibly minimal open chromatin bias. They successfully mapped those dense, silenced domains.
And, even more impressively, they captured bivalent promoters.
Oh, bivalent promoters are fascinating.
They really are. So what does this all mean? Think of a bivalent promoter like a car revving its engine with the parking brake on. It's a gene stuck in a poised state with both active and repressed sticky notes attached to it at the exact same time. It's ready to spring into action the microsecond it gets a signal.
And capturing that delicate poised state across 4 different dimensions is notoriously difficult because chemical bias usually washes out the signal. But CHARM nailed it.
Which is incredible. Let's recap this second subtopic. Bullet 1, CHARM delivers massive high-quality data per single cell. Bullet 2, it maps 3D loops for active genes like SOX2 and repressor blankets for silenced ones like Hox.
And bullet 3, it successfully identifies bivalent genes that are paused and waiting for a signal.
Exactly. So here's a question for you listening. Think about a habit you have that is bivalent, ready to be triggered, but currently held back. What releases your parking brake?
That's a deep question to ponder.
Right. Now, moving on, we know CHARM can map this delicate poised state in a resting cell. But what happens when you throw a grenade into that resting state? I'm talking about cell division.
Mitosis, the ultimate stress test.
When a cell divides, it has to pack up all its DNA, split it, and unpack it. The biological mystery has always been, how does the new cell remember its epigenetic identity during all that chaos?
To investigate this, the researchers used pseudotime analysis. They ordered the single cells chronologically to track their progression through the cell cycle.
And the RNA dynamics were wild. The actual reading of the genes plunges to its lowest point during the M phase, the moment of splitting.
But then, surprisingly, as the new cell enters the G1 and G2 phases, RNA production doesn't just slowly recover. It has two distinct waves of increase.
The mechanics of how the accessibility and histones recover are what really blew my mind, though. They operate on completely different schedules.
Well, completely. Accessibility completely depends on DNA replication timing. If a DNA segment replicates early, it gets its accessibility back first. It's very linear.
But that repressor mark, H3K27Me3, ignores that schedule entirely. 57% of it recovers immediately in the G1 phase, independent of replication timing.
So the question is, how does the new cell know where to put those repressor marks if the DNA isn't even fully rebuilt?
Right. And to help trigger the memory on this, think of a house rebuilding analogy. Chromatin accessibility is like the physical framing of the rooms it has to be built in order sequentially.
I like that.
But the H3K27E3 is like the Wi-Fi signal. It's guided by the 3D spatial proximity. The 3D layout itself helps the cell remember where the silencer marks go.
What's fascinating here is that the 3D organization is basically acting as a physical hard drive for memory during cell division. The space itself is the memory.
It really reframes how we think about biology. Time for another quick recap. Bullet 1, RNA production drops during division but surges in G1 and G2.
Bullet 2, chromatin accessibility recovers sequentially, tied strictly to DNA replication.
And bullet 3, repressive histone marks recover quickly, guided by the 3D folding of the genome itself. Which leads to my next question for you. How much of your own memory is tied to physical spaces? Do you remember things better when you are in the room where you learned them?
I definitely do. Walking into a kitchen makes me remember I was supposed to take out the trash.
Exactly. The doorway effect. So, we talked about this 3D folding acting as a hard drive. Let's look deeper at that spatial architecture because CHARM mapped these 3D genomes at a microscopic 5 kilobase resolution.
That resolution is insane. It allowed them to find the Rabel configuration, which is a specific way chromosomes are anchored, and AB compartments, where active regions segregate from inactive ones.
But how do we know this isn't just random packing? Like, how do we know it's not just genetic spaghetti?
They use some heavy math for that, specifically pair cross-correlation and the db-scan algorithm.
Wait, db-scan?
Yeah, density-based spatial clustering of applications with noise. It scans for dense crowding, separating random noise from actual functional clusters.
And they found roughly 380 discrete 3D clusters of accessible chromatin per cell. These things are tiny, about 110 nanometers wide.
Right, about 1.28 particle radii.
Here's where it gets really interesting. These clusters aren't random. They are filled with superenhancers, mediator proteins, and BRD4.
They basically act like magnetic VIP lounges.
Yes, VIP lounges. Genes inside these 3D clusters have massively higher expression. It proves that linear distance on a DNA strand matters way less than 3D proximity.
If you fold a distant enhancer right next to a promoter in 3D space, they fire together.
Okay, recap time. Bullet 1. CHARM creates incredibly detailed 3D maps of single cells. Bullet 2. The genome forms about 380 active clusters, or VIP lounges.
And bullet 3. These clusters bring distant superenhancers together to boost gene expression.
So, question for you listening. Think about your own workflow. Do you operate better in a linear step-by-step way or by bringing all your resources into one cluster at the same time?
I'm definitely a cluster person. Everything on the desk at once.
Same here, chaos and proximity. All right, so let's move from lab-grown stem cells to applying this to a complex tissue. They looked at the mouse cortex.
Right, moving into the brain. They analyzed 4,416 cells and tracked H3K27SE-ASC, which is a mark for active enhancers.
And the case study that stood out was the GAD2 gene.
Oh, the GAD2 finding was incredible. GAD2 is a gene that is only turned on in inhibitory neurons. And CHARM showed exactly how that exclusivity works physically.
Yeah, it forms this massive cell-type-specific 3D loop to reach an enhancer that is 665,000 base pairs away.
That distance is staggering for a microscopic cell.
It really is. So if we know all this, if we can see these massive loops, can we predict a cell's behavior?
Yes, and they actually did this. They built an advanced regression model combining accessibility, the active enhancers, and 3D interactions.
And they used a math tool called Shapley values, right? Borrowed from game theory.
Exactly. It calculates the exact mathematical credit each biological layer deserves for turning a gene on. And using this, they successfully predicted over 7,000 enhancer-promoter linkages.
And the big revelation from that prediction model was about distal enhancers, the ones really far away.
Yeah, the distal enhancers completely bypass nearby genes entirely. They act like express trains using 3D loops to find their specific targets.
Just flying right past the local stops.
Exactly.
Let's recap this final section. Bullet 1. CHARM works brilliantly in complex tissues like the brain. Bullet 2. It revealed how distant enhancers, like for GAD2, loop across huge distances.
And bullet 3. Using machine learning, scientists can now predict gene expression based on 3D epigenetic shapes.
So, listeners, if scientists can predict what a brain cell will do based on its folding, how might this change the way we treat genetic diseases in the future?
It's going to change everything.
It really is, because CHARM isn't just a niche lab tool, but reading 4 layers of biological data at once, we are moving from looking at blurry, flattened maps of biology to full 3D interactive holograms.
Absolutely. This is the foundation for highly personalized medicine. But this raises an important question. What are the current limitations?
Right. We have to be realistic.
Exactly. Because of the intense detail per cell, the method is currently low throughput, but it absolutely paves the way for future automation. And imagine what happens when we add even more layers, like DNA methylation.
The hologram is only going to get sharper. So I want to leave you, the listener, with a final thought.
Let's hear it.
We spend so much time thinking of our bodies as biological machines driven by fluids and electricity. But if our cells are constantly unpacking and refolding their DNA in 3D space just to remember who they are our bodies actually incredibly advanced physical hard drives.